Hello everyone. So from today onwards, we are going to start a series of MCQ solving. So first, we are dealing with the microbiology, general microbiology. So MCQ solving is very important in such a volatile subject. So let's start. Microorganisms used as weapon in biological terrorism. Smallpox, rabies, influenza, and human parvo. So, among these, the potential weapon which can be used is a smallpox virus. Option A, smallpox is correct. Diseases transmitted by blood transfusion except syphilis, blood transfusion, AIDS, blood transfusion, malaria, blood transfusion, histoplasmosis is a fungal infection. So, it is by inhalation of spores. It is by inhalation of spores. It is by inhalation of spores. All others, syphilis, AIDS, and malaria, transmitted by blood, except the histoplasmosis. Correct answer is histoplasmosis. True about sterilization. Sterilization, spores are destroyed by drying. Yes. Soil rusting is destroyed by hot air. No. Spores is destroyed by drying. No. Spores cannot be destroyed by drying. Autoclaving should be used to destroy. Best method is autoclaving. Is it sterilized by hot air oven? No. Surgical instruments? No. Boiling cannot be because spores will not be then destroyed. Vaccine sterilized by filtration? Yes. The answer is vaccine sterilized by filtration. Next question. The following statements regarding bacterial spores are correct. Except which one is incorrect? We have to find out. Are spores are formed under adverse environmental conditions? True. Resistant to boiling? Yes. Contain multiple layers? Spores? Yes. Are formed primarily in the organism genus not Streptococcus. Streptococcus do not form spores. Two genus that form spores are Bacillus cereus, Bacillus anthrax. Other is Clostridia species, Clostridium perfringens. Clostridia species. These two form the spores, Bacillus and Clostridia. Next question. The chemical nature of capsule, maybe chemical nature of capsule is a polysaccharide. In majority is polysaccharide. Some of this is polypeptide as seen in anthrax. So the correct answer will become both of them. Now the next question is most important surface active disinfectants are. So surface acting disinfectants are cationic. They are cationic. The purpose of staining cells on a microscopic slide. Staining is to get the clarity, right? Contrast in order to see them better. Fixation is to kill them and secure them to the slide. So for fixation we do kill them and secure them to slide. But to contrast them, see better, we use staining. A microbiologist inoculates Staphylococcus epidermidis and Escherichia coli in a culture medium. Following incubation, only Escherichia coli grows in culture. What is the most likely explanation? You use too much inoculum? No. This culture is contaminated? No. The incubation temperature was incorrect. The culture medium was selective. It must be selective. Because of that, E. coli is growing and Streptococcus epidermidis is not growing. Staphylococcus epidermidis because it is like a normal flora. Next question. You're observing a cell through a microscope. No, it has no apparent nucleus. No apparent means it do not have nucleus. Nucleus is absent. You conclude that it is most likely to be. So it is most likely to be a bacteria, and bacteria have peptidoglycan cell wall. Cellulose for fungus. So here we have peptidoglycan because it do not have nucleus, so it's a bacteria. Moves by pseudopodia is by protozoans like amoebas. Multicellular animals, so it is animals. No. But here no nucleus, so it will be peptidoglycan cell wall bacteria. Next. Let's see next question. So next question is Coast postulates are criteria used to establish that microbes are found in dust particles. A specific microbe is the cause of a specific disease. Learn all the postulates. Life forms can only arise from pre-existing life forms. This is not by coach. A specific microbe should be classified as specific kingdom. No. Microbes can be used to clean up toxic species. Not. Coach just talked about the specific microbe is the cause of a specific disease. Let's see next question. All microorganisms are best defined as organisms that cause a human disease? No, all microorganisms do not cause. Some are very useful also, like our gut bacteria, which live symbiotically with us. Lack a nucleus. Just they lack a nucleus. Are infectious particles are too small to be seen with the unaided. The correct definition is the microorganisms with which we cannot be seen with the unaided naked eyes because they are too small. Micro means too small organisms that cannot be seen with the naked eyes. So the correct answer is D. Can only found growing in laboratories? No. Which is true for endotoxin. Endotoxin highly antigenic only in gram negative bacteria. Yes, it is found only in gram negative bacteria because gram negative bacteria only have what? Find out and write in the comment section. Which subunit of the exotoxin is the binding portion of the molecule? Binding portion is BB for binding. Okay. Let's talk about the next question. Gram negative bacteria have a lipopolysaccharide which is found in which membrane? Which is found in the outer membrane. Which is found in the outer membrane. So outer membrane have lipopolysaccharides, and lipopolysaccharides are responsible for endotoxins. That's why only gram negative bacteria can produce endotoxin, not gram positive, because of the outer membrane. Outer membrane have lipopolysaccharide. Probiotics exhibit all but the following characteristics: non-toxic and non-pathogenic, a beneficial effect on host, mimic antibiotics. Yes, they help us, right? Remain viable for long time, withstand acidic nature of the host stomach. They cannot withstand. No, 15 is yes. Some are living in stomach also. Yes, remain viable for long time. No, they cannot remain viable for a long time. The following statement is true. Blood culture should be collected when the patient spikes a fever. When the patient spikes a fever. Blood culture should be collected after the patient spikes a fever. Blood culture should be collected before the patient spikes a fever. Blood culture collection need not coincide with the fever spikes. Anytime you can collect the blood. Phenol is a bactericidal at the concentration of. So phenol is a bactericidal at the concentration of 1%. 1% you can kill the bacteria. So these are the important questions which need to be studied well. So lipopolysaccharide is responsible for endotoxin in gram negative. So the correct answer is 1%. So 1% phenol is used as a disinfectant to kill the bacteria, and 1 to 2% if you take, then you can kill the fungus also. So now let's see 18th question. Which of the following is the most ideal disinfectant for preparation of skin prior to insertion of central venous catheter? Central venous catheter is a very important and you have to take precautions. Disinfection should be proper. So what you can use? You can use chlorhexidine, that is 2% weight by volume chlorhexidine you can use. That will that will disinfect the skin. Bowie Dick test is used to assess. So Bowie test is used to assess for steam sterilization. The performance of steam sterilization can be noted. Performance of steam sterilization can be noted by Bowie Dick test. About the sterilizer. Okay. The spore of the following is used as a biological unit to monitor steam sterilizer. For steam sterilizer, which one you have to use? So for different you have to use different. Subtilis for the hot air oven. For this one you have to use the Geobacillus stereothermophilus. This one, yeah, first one, that is Geobacillus, you have to use for monitoring steam sterilizer. So it should be stereothermus. Thermophilus. The following phenomena is responsible for antibiotic resistant bacteria due to slime production. Antibiotic resistance due to slime. Slime forms a biofilm. Biofilm formation, such as in Streptococcus epidermidis on the um, implants, so it will form. So that will protect it from antibiotics. That is slime production. Because of that, it is getting antibiotic production protection. Okay, now let's discuss universal standard precautions should be applied to all except. So the universal or standard precaution should be applied to all except semen, synovial fluid, sputum, none. So, but now sputum and blood. These two are for the universal standard precautions. The first thing to do in case of accidental occupational exposure to blood. So, if there is any accidental exposure to blood, then what you have to do? Wash with plenty of soap and running water. Yes, you have to do this. Put the affected area? No. Never keep in mouth. Squeeze blood from the area? No. Apply bleach. Never add irritants to your affected area. Always just wash with soap and water and go to the and go for evaluation, like reports of the patient, whether they are HIV, if any blood test available, your blood test, and and if you have not vaccinated, then go for the vaccine and for the uh, medicines, right? How should endoscopes be disinfected? How to disinfect the endoscope? The manual cleaning is done. Manual cleaning, disinfecting, and rinsing. Okay, see, the this one you can do manual cleaning. So what is used for disinfecting the endoscopes? So you can use 2% glutaraldehyde for the manual cleaning of endoscopes. Okay, 2% glutaraldehyde. 2% glutaraldehyde. 2% glutaraldehyde you can use. In a post-operative care unit, 5 patients develop post-operative wound infection on the same wound. The best method to prevent cross-infection occurring in other patients in the same ward. So the best method is following hand hygiene. So practice proper hand washing. Okay, that will prevent the cross-infection. Okay, cross-infection can be prevented by proper hand hygiene. Endoscopes have been known to be contaminated with the following organism due to poor quality of rinse water. Rinse water sometimes they use, most of the time, that is Mycobacterium chiloni. So Mycobacterium chiloni is the most common for the rinse, because of the rinse poor quality of the water which is used for rinsing the endoscopes. Now, peptidoglycan synthesis occurs in 4 phases. Cross-linking of polypeptide chain occurs in which phase? So this is a quite remembering topic. So this is phase 4. Last phase, in last phase you will get the cross-linking. Okay, cross-linking of a NAG and NAM. What is the structure that is found in gram-negative but not in gram-positive bacteria? Gram-negative but not in gram-positive bacteria. What is the structure that is found in gram-negative but not in gram-positive bacteria? What is the structure found in gram-negative but not in gram-positive bacteria? Okay. So, what is the structure found in gram-negative but not in gram-positive bacteria? See, gram-negative bacteria but not in gram-positive. So, gram-negative bacteria, both of them have cell wall. Both of them have plasma membrane or cytoplasmic membrane. Both of them have flagella. But gram-negative have a special thing, that is outer membrane. Outer membrane is absent in the gram-positive bacteria. My pen is not working now. Okay, fine. So gram-negative bacteria has a special structure, that is outer membrane. Okay, exotoxins. So exotoxins are lipopolysaccharide in nature. No, produced by gram-negative bacilli. It is produced by both. Highly antigenic, yes. It is very stable and resistant to chemicals. No, it is not very stable. You can toxify it. If you toxify it, then what will happen? They will become a toxoid, and when toxoid is there means you can use it for vaccination. So mostly exotoxins are produced by gram positive bacilli, mostly. So you can go for option C. Gram positive also produce, but the most specific thing is highly antigenic. Okay, this is the thing. So talking about the previous question, which linkage was there? So see here, uh, peptidoglycan is also known as murin. So this peptidoglycan is also known as murin. So this murin is a polymer. Peptidoglycan is a polymer of sugar and amino acids. It is a polymer of sugar and amino acids. They form a mesh-like structure, mesh-like layer outside the cytoplasmic membrane, that is plasma membrane, of most bacteria, forming the cell wall. The sugar component consists of alternating residues of beta 1,4 linked and acetyl glucosamine, beta 1,4 linked and acetyl glucosamine NAG, and it also have NAM. So NAG and NAM, N-acetyl glucosamine and acetyl muraminic acid. So attached to N-acetyl muraminic acid is a peptide chain of 3 to 5 amino acids. So 3 to 5 amino acids are joined to it. The peptidoglycan chain can be cross-linked to the peptide chain of another of another strand, forming the 3D mesh-like layer. Peptidoglycan serves as a structural role in the bacterial cell wall, giving structural strength, as well as contracting the osmotic pressure of the cytoplasm. Okay, peptidoglycan is also involved in binary fission during bacterial cell wall production. Okay, so these exotoxins are highly antigenic. It is produced by both gram negative as well as gram positive, not just by gram negative but by both gram positive as well as gram negative. It's highly antigenic, toxidable. You can weaken it by heating, and it is heat level. It's not very stable. Let's talk about the next question. A soldier developed thigh abscess following gunshot injuries during an anti-insurgery combining Operation Gram stain of the foul smelling. So the foul smelling exudate is there. Foul smelling exudate revealed plenty of pus cells and a mixed flora of gram positive, gram negative rods. However, aerobic culture was sterile. How was the aerobic culture? Aerobic culture was sterile. Sterile. Which of the following is most likely involved in the following reactions? Which of the following enzymes is most likely involved in the following reactions? This is the reaction, guys. This is the reaction. So which of the following enzymes is most likely involved in the following reaction? So here you can see this is a superoxide. This is a superoxide, and it is giving peroxide and oxygen and peroxide. So it will be superoxide dismutase. So superoxide dismutase, it is also known as erythrocyprin. It catalyzes the reaction of two molecules of superoxide to yield one molecule of hydrogen peroxide, two molecules and one molecule of hydrogen peroxide, and one molecule of oxygen. Diffusion of hydrogen peroxide, the product of SO_D1 across the cytosol is limited. Okay, so to yield one molecule of hydrogen peroxide and one molecule of oxide. Diffusion of hydrogen peroxide, the product of superoxide dismutase 1 across the cytosol is limited. Okay, now let's see next one. On gas pack, two types of bacteria were isolated. Biochemical studies reveal that these bacteria fail to produce superoxide dismutase catalase and peroxidase. All are incorrect except Okay, so what it is saying on gas pack, two types of bacteria were isolated. So biochemicals revealed that these bacteria failed to produce superoxide dismutase, catalase, and peroxidase. These three all are incorrect except aminoglycoside antibiotic is the drug of choice, less virulent than the ones containing all three enzymes. The enzymes are expressed in the reduced state. They have enzymes only for EMP pathway. So these enzymes are very much essential. So these are present only for the EMP pathway. So an important route of glucose conversion to pyruvate such as glycolysis, that is Embden-Meyerhof pathway. Glycolysis is the most important type of mechanism by which organisms obtain energy. So for that, from organic compounds in absence of molecular oxygen, as it occurs in the absence of oxygen, therefore it is called anaerobic fermentation. So for anaerobic fermentation, it is required. All are true regarding disinfectants except. So disinfectants, we're going to talk about disinfectant. So all are true regarding disinfectants except. So glutaraldehyde is sporicidal. Okay, yes. Hypochlorites are virucidal. Yes. Ethylene glycol is intermediate disinfectant. Yes. Phenol usually requires organic matter to act. Actually, it acts in the presence of organic matter. Organic matter is much needed. That is not true. So this is wrong. These three are correct. Glutaraldehyde is sporicidal to kill the spores. Hypochlorites, they can kill viruses. Ethylene glycol is intermediate disinfectant. What is the example of intermediate disinfectant? Glutaraldehyde. Glutaraldehyde. Now let's discuss this. Endoscopes have been known to be contaminated with the following organism due to poor quality. This we have done, right? This is Mycobacterium cheloni. Mycobacterium cheloni that is present in the tap water and that contaminates endoscopes. Which of the following is used for biological monitoring of gas plasma sterilizer? So gas plasma sterilizer, for that you have to take some bacteria and check for it. There is Bacillus stereothermophilus. Okay, so which of the following is biological monitoring? That is Bacillus stereothermophilus. After this, you can use Bacillus atrophius subspecies niger, and after this, you can use Bacillus pumilus radiation. Okay, now let's see the next question. The best way to prevent contamination of the PCR process. So how can you prevent the contamination of PCR process? Use of biosafety cabinet? No. Internal sterilizing agents such as uracil and glycosyl? Yes. Use universal precautions, even then it will get contaminated. Clean the area with 1% sodium hypochlorite. It's not very good. Okay, so the most common strategy to prevent carryover contamination is to partially or completely replace DTTP with DUTP during PCR amplification, thereby producing DNA containing uracil. So prior to initiating PCR, the PCR mixture is treated with uracil DNA glycosylase UNG glycosyl. Okay, during the initial denaturation process. So during the initial denaturation step, temperature is elevated to 95 degrees, resulting in cleavage of a pyridinic site and fragmentation of carryover DNA. Okay, then after that, as the template contains thymidine, it will not be affected by UNG treatment. It is prerequisite that all PCRs are carried out with DUTP substituting DTTP. Okay, so okay, so you can say that that is UNG is required. Next question: the best molecular typing method is. The best molecular typing method is PFGE, that is gold standard for genotyping tool. For genotyping, for molecular typing tool. Other genotyping methods are chromosomal DNA analysis, plasmid profile analysis, RFLP, that is restricted fragment length polymorphism, then ribotyping, that is RFLP analysis by ribosomal DNA. Okay. All except one are contributions of Robert Koch to the field of microbiology. All except one are contributions of Robert Koch to the field of microbiology. All except one are contributions of Robert Koch to the field of microbiology. So, what is that? A germ theory of disease. A germ theory of disease. All except one are contributions of Robert Koch to the field of microbiology. One are contributions of Robert Koch to the field of microbiology. All except one are contributions of Robert Koch to the field of microbiology. So, all except one are contributions to the field of microbiology. So, germ theory of disease? No. It was not given by Robert Koch. Discovery of Vibrio cholera? Yes. He discovered Vibrio cholera. Staining technique. Staining of bacteria. Discovery of solid media. Koch discovered solid media. So these are all regarding the Koch, but the germ theory of disease is not from Koch. Okay. Next question. The Paul Ehrlich is known for the contributions to microbiology in the field of Paul Ehrlich. So Paul Ehrlich, discovery of protozoals as antibacterial, discovery of gene splicing, theories of immunity. Yes. In the field of theory of protozoals. In immunity, theories of immunity, theory of antibiotic production, no. Theories of DNA synthesis, no. So, Prontocil was given by Domag. This was given by Domag. Domag. Okay. And genes theory, Journey's theory of antibody protection using the concept of clonal selection. That this antibiotic protection theory of clonal product selection, it was given by Journey's theory. Okay. Okay, this is not very much required. Okay. Paul Ehrlich, he gave the theories of immunity. Peptidoglycan synthesis occurs in 4 phases. Cross-linking peptidoglycan occurs in, so it occurs in the last phase, that is phase 4. It occurs in the last phase, that is phase 4. Done. Sedimentation coefficient of bacterial chromosomes. Bacterial chromosomes, they have 70s and 30s. 70s is the correct option. 70s. So it is 70s. It is made up of 30s and 50s. But humans having 40 plus 60. Yes. We have 80s. Okay. Now, biocides are substances that. So, I want to tell one concept here. So, based on this, we can, based on the bacterial chromosome, we can get some knowledge about which antibiotic, how we can use it. So, antibiotic resistance is natural and acquired. Acquired is when there is a mutation and gene transfer. And there is a mutation and when there is a gene transfer. Gene transfer, you can have vertical and horizontal. Vertical and horizontal. In horizontal, conjugation, transformation and transduction. And these vertical, generation to generation by reproduction. And horizontal is by conjugation, transformation and transduction. I hope you understood this. Biocides are substances that kill only pathogenic microorganisms. No. Kill all living microorganisms excluding spores. No. Kills all living microorganisms including spores. Yes. Prevents growth of pathogenic. No. So kills all living microorganisms including spores. So cidal means to kill. Kill. Cidal means kill. Tactic agents means to prevent the growth. Cidal means to kill all the microorganisms including the spores. Biocide. Virucidal means to kill the viruses. Chemical agents that destroy the microorganisms by interfering with the membrane functions are interfering with membrane functions. Chemical agents that destroy the microbe by interfering with membrane functions are chemical agents that destroy the microorganisms by interfering with membrane functions are phenol. Is phenol. Formic acid? No, no, no. Ethylene oxide? No. Phenol. So phenolic agents, they act by interrupting the cell membranic functions. Whatever cell membrane functions are there, they interrupt it. Phenolic agents. Okay, that is interfering the membrane. Ideal concentration should be 5%. 5% phenol. Ethylene oxide and hydrogen peroxide acts on DNA. This ethylene oxide and hydrogen peroxide, they are very strong, so they act on the DNA directly. Aldehydes are strong alkylating agents. These aldehydes are strong. These formaldehydes, formic acids, alkylating means they also interfere with the DNA itself. Okay. Next question is not true about drug resistance mechanism. Not true about drug resistance mechanism. So most common mechanism production of neutralizing substances. So most common mechanism is production of no. If resistance is plasmid mediated, it is always transferred vertically by division from generation to generation. Not true about drug resistance mechanism. It's not true. This is not true. It is vertical, not vertical. It is horizontally. It is horizontally transferred. Plasmid mediated, not vertical, horizontal. Alteration of target in pneumococcal resistance. Yes. Complete removal of target is cause of resistance to vancomycin. Most common mechanism is production of neutralizing substances. So B is wrong, not true, because it is not vertical, it is horizontal. So plasmid mediated gene transfer like conjugation. Conjugation is horizontal gene transfer method. Horizontal gene transfer method. Plasmid mediated. Okay. Next question. Let's do the next question. As we have discussed now, gene transfer is vertical and horizontal. Vertical means from generation to generation by reproduction. Horizontal means conjugation, transformation, transduction. Let's talk about all the following are required to in PCR except which one is not required for PCR. Deoxyribonucleotides required. Thermostable except we have to find out. Thermostable enzyme or DNA polymerase very much required. Deoxyribonucleotides. Ribonucleotides की क्या जरूरत है? PCR. Magnesium and single stranded DNA, template DNA. Yes, it is required. Template DNA or single stranded DNA is required. The important thing need for PCR are DNA templates. Template is required. Once you have template, then only you can make. DNA polymerase. Polymerase is required. Primers are required. All primers are required. Primers. Nucleotides are required. Then only new DNA will form now by taking the nucleotide. And reaction buffer. Buffer is required. These things are required. 5 things. Template should be there. Polymerase enzyme should be there. Primer should be there. Nucleotides and reaction buffer. Penicillium is extracted from. So Penicillium, Chrysogenum notatum, Aspergillus niger and yeast. These are all fungus, no doubt. But penicillium, that is penicillin. Penicillin is extracted from Penicillium chrysogenum and Penicillium notatum. These two are the good options. Notatum से penicillin आ जाएगा and chrysogenum से आ जाएगा. Penicillium species से you will get penicillin. These two fungus. Okay. From Aspergillus niger you will not get penicillium. So for some extra information, Penicillium chrysogenum, you will get penicillin. From Penicillium fulvum, you will get grisofulvin. And Acrimonium species, you will get cephalothin. So different bacteria, different things you can get. So which of the following statements are true regarding flagella? So let's talk about flagella. Flagella is a very important structure. So let's talk about flagella. So flagella made up of smaller units called flagellin. Yes, true. It is asking true regarding. So we got first option. So bacteria with polar flagella move fast. एक ही साइड पे flagella है, it cannot move. Because more number of flagella or more. Only one flagella, it cannot move fast. Antibodies to flagella are protective. To flagella, they are productive, not protective. Basal body is anchored in cytoplasm. So the flagella contains tail, hook, and basal body. Not it is not anchored in. It is present outside. All flagella essentially have same chemical composition. All are true. It is asking. A, B, and E is the correct answer. This is also true. Have same chemical composition. All flagella, they have same chemical composition. Okay, one side flagella is there, then also polar flagella, bacteria can move fast. Okay, fine. So these are the questions. Let's talk about some important things like lysogenic cycle. So next question is lysogenic cycle. So it's like more than one option is correct. Lysogenic conversion is seen in all for the following. Lysogenic conversion is seen in how many? Clostridium diphtheria, yes. Salmonella, yes. Clostridium botulinum, yes. So in these three you can see lysogenic conversion seen in cholera toxin, Vibrio, cholera toxin, Salmonella O antigens, Clostridium botulinum, and exotoxins A to C, that is atherogenic or pyogenic, that is of Streptococcus pyogenes. Corynebacterium diphtheria species is Shiga toxins. Corynebacterium diphtheria toxin and Shiga toxin. So you will get A, B, D, E. Multiple correct answers are there. A, B, E. So Vibrio toxin, that is Vibrio cholera, not just Vibrio cholera, E you will get. So don't choose this option because cholera is not good. Okay. So Corynebacterium diphtheriae toxin, Shiga like toxin, and Salmonella O antigens are there. Not Streptococcus, you can see Streptococcus pyogenes A to C exotoxins, they have a lysogenic cycle. Corynebacterium diphtheriae and Clostridium botulinum. Shiga like toxins also you can see cholera, Shiga like toxins, cholera toxin, Shiga like, and Salmonella O antigens. Salmonella O antigens you can see Clostridium botulinum, Corynebacterium diphtheria, and Streptococcus. So cholera toxin, Shiga like toxin, not Shiga like, Shiga toxin, cholera toxin, Shiga toxin, and Clostridium botulinum toxin, Corynebacterium diphtheria toxin, Clostridium botulinum and Corynebacterium diphtheria. Then you can have Salmonella and then Streptococcus. Salmonella O antigens. Now let's talk about this one. Which of the following examples of basic dyes used to stain bacteria? Used to stain bacteria. Which of the following are examples of basic dyes used to stain bacteria? So basic dyes which are used to stain bacteria. Crystal violet, of course, we use crystal violet a lot. Methylene blue also we use, and saffron also. saffron, malachite green also. So these all we use. Okay. Basic dyes, crystal basic dyes. These dyes have positive charge and bind to negatively charged molecules like nucleic acid. Okay, COOH and OH groups. They bind to these molecules. They are basic dyes. They have positive charge. The basic people are positive. Since surface of bacterial cells की जो surface है it is negatively charged. Since surface of bacterial cells are negatively charged due to teichoic acid, basic dyes are most commonly used in bacteriology. Surface of bacterial cells क्या रहते हैं? Negatively charged due to teichoic acid. So basic dyes are commonly used in bacteriology because basic dyes are positively charged, so they bind to negatively charged. And the surface of bacterial cells are negatively charged due to presence of the teichoic acid. Basic dyes are most commonly used in bacteriology. Example: crystal violet, methylene blue, saffron, basic fuchsin, malachite green. Okay, so this is the multiple correct answers are there. Molecular technique used to study expression of gene is molecular technique used to study the expression of genes. Polymerase PCR no, real PCR no, ligase reaction no. Yes, microarray using gene chip can be used to study molecular technique used to study expression of genes. So chip can be used to study the expression of genes. A microarray actually it is a laboratory tool. It is used to detect the expression of thousands of genes at the same time. DNA microarrays are microscopic slides that are printed with thousands of tiny spots in different positions, with each spot containing a known DNA sequence or gene. So let's start with other question. Following statements are true regarding real-time PCR. So real-time PCR is also called PGI. So following statements are true regarding real-time PCR. So uses fluorescent probe for detection of amplified product. Yes. Post amplification manipulation is required. Cycle is very long. Risk of release of amplified product into the environment is less. Yes. Quantification cannot be done. So which one is true? So these are true. Uses fluorescent probe. Yes, it uses fluorescent probe. And yes, it uses a fluorescent probe. So yes, it uses the fluorescent probe. And risk of release of amplified product into the environment is very low. So mycin is next question, a bactericidal antimicrobial. So mycin is bactericidal antimicrobial. It is bactericidal antimicrobial. Effective against gram-positive bacteria. Yes, sure. Effective against gram-negative. Yes. So mycin is a bactericidal antimicrobial. Antimicrobial antibiotic है ये. Not pneumonia. No. So they are poorly absorbed from GI and cannot be given orally. They have to penetrate the spinal fluid poorly. So they penetrate the spinal fluid poorly and not suitable for treating meningitis. You cannot treat pneumonia. Active even during stationary phase of growth cycle. So then perfect answer is it is a bactericidal antibiotic against all gram positive negative, but because it cannot cross the lipid membrane, that's why it is not useful for pneumonia, meningitis, all these. Okay. Which of the following is or are examples of live vaccines? It's very important. Live vaccine कौन से हैं? These are asked a lot of time. Which of the following is or are example of live vaccine? So live vaccine is example BCG vaccine and oral typhoid vaccine. Oral typhoid vaccine and BCG vaccine. These are the live vaccines. BCG and typhoid vaccine. Oral typhoid. Japanese encephalitis killed vaccine. Nakayama strain and attend live vaccine strain. Influenza is live attenuated Live attenuated nasal spray, not in routine use. You cannot take that. So influenza virus का live vaccine, but live attenuated vaccine, but it is not used. So remember, BCG and oral typhoid vaccines are the live vaccines. Are the live vaccines. Following statements are true regarding external quality assurance of EQA. Which one is the assurance? So establish intra laboratory performance, no. Ensure reliability of future testing, yes. Quality is required for ensuring the future testing, right? Reliability. Ensure credibility of the laboratory, yes. Retrospective periodic assessment, yes. Establish laboratory comparability, yes. External quality assessment is EQA. EQA is the challenge of the effectiveness of a laboratory quality management system. EQA typically refers specifically to these challenges, to the challenges of quality system in a medical laboratory. The term external refers to the fact that an organizer outside of the laboratory's organization provides a statement of quality to the laboratory. Next, ophthaldehyde has following advantage over glutaraldehyde. So orthophthaldehyde has following advantage over glutaraldehyde. What is that advantage? It is non-irritant. It do not irritate like glutaraldehyde, and other is it is odorless. It do not have bad odorless. So OPA, that is orthophthaldehyde, has the advantage that it is a solid. It is a solid compared to liquid glutaraldehyde, though both are normally supplied. It is a solid. Which of the following statement is true? Next question, which of the following statement is true? Which of the following statement is true? A solid media are enriched media. It is telling you solid media is the enriched media. Nutrient broth is a basal media. Nutrient broth is a basal media. Agar adds nutrients to media. Never. Agar do not add nutrients to media. Never. Chocolate agar is a selective media. No, it is not selective. Nutrient broth is a basic media, basal media. Solid media enrichment, no. Liquid can also be enrichment. So 55B is the right answer. Nutrient broth is basal media. Which of the following is most resistant to antiseptics? Antiseptics के लिए सबसे ज़्यादा resistant कौन रहता है? Prions. Prions is the right answer. Prions are most resistant to antiseptics. Least resistant is enveloped viruses. More resistant is prions. तो ऑर्डर कैसा रहता है? पहले वायरस है जो कम रेसिस्टेंट है, वो जल्दी मर जाते, बाहर बॉडी के बाहर ज़्यादा देर तक वायरसेस नहीं रहते हैं. फिर इसके बाद ग्राम पॉजिटिव बैक्टीरिया, देन नॉन इन्वेलप्ड वायरसेस, फिर उसके बाद नॉन इन्वेलप्ड वायरसेस, इसके बाद फंगाई एंड फंगल स्पोर्स, फिर ग्राम नेगेटिव बैक्टीरिया, देन आफ्टर दैट प्रोटोजोन ट्रोफोजोइड्स, फिर cyst are very resistant, then staphylococcus and pseudomonas, then you have mycobacterium, then you have bacterial endospores, then you have prions are more resistant. They are more resistant. Okay, which of the following is most resistant? That is prions. Most resistant is prion. But among this, if you want to write the order, then you can write as write as fungus, then cyst, then spores, then prions. Okay. A chest physician performs bronchoscopy in the procedure room of the outpatient department to make the instrument safe for use in the next patient waiting outside. What do you have to do? What do you have to do? So, what you have to do is, as I discussed in my previous, you have to use glutaraldehyde. How much percentage? 20%. 2%. 2% for 20 minutes. 2% for 20 minutes. Ideal method: disinfecting heat labile equipment like endoscopes are 2% glutaraldehyde for minimum 20 minutes. Contact period should be there 20 minutes. सोडियम हाइपोक्लोराइड स्पिल्स मैनेजमेंट के लिए यूज़ होता है. सोडियम हाइपोक्लोराइड स्पिल मैनेजमेंट. Formaldehyde for fumigation and ethyl alcohol for normal alcohol for stethoscope. Formaldehyde for fumigation, sodium hypochlorite for spill management. If there is a blood spill, any spill or chemical spill, you can use sodium hypochlorite for blood spills management. 2% glutaraldehyde for 20%. See, solid media are enriched media. Solid media, they are not. Liquid media can also be enriched media. So mostly the enriched media are liquid media only, not solid. Liquid media are enrichment media. Enriched is different, enrichment is different. Liquid media are enrichment media. Agar it do not provide any nutrient nutrition. Chocolate agar is selective, it is not selective, it is enriched. It is enriched with chocolate. Enriched. Remember I'm telling. Okay, next question. So we have discussed a lot of question by now. Heat labile instruments for use. Heat labile instruments for use. Heat labile instrument for use in surgical procedures can be best sterilized by. Heat labile भी है and surgical procedure के लिए heat labile instrument for use in surgical procedure can be best sterilized by ethylene oxide gas you can use. So ethylene oxide is sterilant. Any surgical equipment contact with the blood or tissue is comes under critical, so it need to be sterilized. In case of heat labile atoms, ETO ethylene oxide is the ideal method. Next question. All are true regarding disinfectants except स्पोरिसाइडल. यस ट्रू है. ट्रू पहचान और जो नहीं है ट्रू एक्सेप्ट ट्रू. हाइपोक्लोराइड्स आर वायरुसाइडल. यस यू कैन से दैट. इथिलीन ऑक्साइड इज इंटरमीडिएट डिसइंफेक्टेंट. ओके नॉट ओके. Phenol usually requires organic matter to act. This we have already discussed. It require in the presence of organic matter it acts, but it doesn't mean that organic matter is needed for the action of phenol. So ETO is not a disinfectant, it is a sterilant. It is a sterilant, not a disinfectant. Okay, it is a sterilant. So both the two options are incorrect. The operating temperature in an ethylene oxide sterilization during warm cycle should be around 49 to 63 degrees Celsius. The warm cycle of ETO 30 to 65 degrees Celsius for about 8 to 12 hours. Choose the correct ones for the following. For the decreasing order of resistance to sterilization. So first, as we have done in previously, decreasing order we have to use. सबसे ज़्यादा prions का था, no doubt. In what is first option में prions you can choose. Then bacterial spores will come. Then bacteria will come. Okay, next let's discuss next. Sterilizing agents include. So sterilizing agents, they include dry heat, ETO. Alcohol is not a sterilant, disinfecting. Ether disinfecting, chlorhexidine disinfecting. So two options are correct, A and B. ETO, ethylene oxide and dry heat. So the following can be reliably used for hand washing. for hand washing. Can be reliably used for hand washing के लिए which one you can use? You can use chlorhexidine and isopropyl alcohol. These two agents are reliable agents for hand washing. Anionic agents like detergents and soaps are also useful agents in hand washing. Soaps, the anionic. Okay, next question. Sporicidal agents are sporicidal, spores को kill करने वाले agents कौन से हैं? Glutaraldehyde, yes, sure. ETO, it is a sterilant, so it will definitely kill. And formaldehyde. ETO is very effective with sporicidal agent. Aldehyde is activated stage. In activated stage, they exhibit sporicidal activity. Radiation can be used to sterilize all except radiation. Say you can use bone structure and artificial tissue graft. Bronchoscopes, no. Manually you have to clean, or you have to use ETO for 20 minutes. 2% glutaraldehyde, not ETO, glutaraldehyde for 20 minutes. Sputum disinfection is done by all except. Sputum disinfection कैसे करते हो? By all except autoclaving से sputum disinfection कर सकते हो, क्रिसॉल से कर सकते हो, boiling you can do, but what about chlorhexidine? No, chlorhexidine you cannot. Sputum is disinfected by using 5% phenol, cresol, autoclaving, boiling and burning. This is chlorhexidine. Which of the following is most resistant to action of antiseptic and disinfectants? Most resistant to action of antiseptics and disinfectant, as we have discussed, prions are the highest. Okay, most resistant, and least are enveloped viruses. The indicator used in autoclave is so autoclave or steam sterilizer you can use Geobacillus stereothermophilus. Biological indicator in autoclave is Geobacillus stereothermophilus. And for radiation you can use Pumilus radiation, Bacillus pumilus. And subtilis or atrophius you can use for hot air one. Hot air के लिए subtilis you have to use. Bacillus subtilis for hot air. ये मीडियम टेम्परेचर कर सकता है, बहुत ज़्यादा भी नहीं कर सकता. ओके, विच इज़ यूज्ड इन डाइजेशन एंड डिकंटामिनेशन स्पूटम इन स्मीयर प्रिपरेशन. Used in digestion and decontamination of sputum in smear preparation. That is sodium hydroxide is used as digestive as well as decontaminating agent in sputum sample processing for smear preparation and culture. And sodium hydroxide method is currently employed in many laboratories. Which of the following are used for sterilization of surgical instruments? For surgical instruments, ethylene oxide, ETO, gamma radiation, autoclaving, glutaraldehyde, hot air oven, all the above you can use for disinfecting surgical instruments. So a surgical instrument is a broader term actually, which includes scalpel, blades, suture material, many other items. All the choices are used for different typical surgical equipments. Okay, different equipment, different is used. The disposable plastic syringes are best sterilized by. So best it can be by ETO. ETO disposable plastic syringes. Autoclaving is done by. How can you use autoclaving? How can you do? So there's a specific temperature that is 121 degrees for 15 minutes. For 15 minutes at 15 lb pressure. C is the right answer. 15 minutes for 15 lb pressure. 121 डिग्री सेल्सियस for 15 minutes 15 lb. 15 मिनट में आपका स्टीम स्टरलाइजेशन हो जाएगा. That is autoclaving. The steam at 100 degrees for 30 minutes is for Koch Arnold steamer. And dry heat के लिए 160 degrees for 30 minutes. Or 120 minutes. Not actually 30, 120 minutes. For dry heat, dry air. That is for hot air oven. hot air oven 160 degrees for 120 minutes. Culture media are sterilized by culture media. Okay, so culture media these are sterilized by autoclaving. Culture media किस तरह से sterilize करते हो? By autoclaving. तो hot air oven like oils, glassware, liquid, paraffin, metals you can use. Tindelization other names are intermittent sterilization or fractional sterilization. So culture media sterilized by autoclaving. Autoclaving you can use for sterilizing culture media. Glassware and syringes. Syringes these are sterilized by hot air oven. Glassware and glass syringes are sterilized by hot air you can use. Plastic syringes by gamma radiation. Heart lung machine by ETO. Ventilator by ETO. Okay. Glassware किससे करना है? Hot air oven. Glass can bear the hot temperature. Hot air. Okay. Asepsis means asepsis क्या होता है? What is asepsis? Absence of pathogenic microbes. Yes. Disinfection no. Disinfection में spores रह जाएंगे, virus रह जाएगा, prion रह जाएगा. No. Prevention of infection no. Destroying all forms of microorganisms. All forms is not required. Pathogenic चले गए, बस इतना ही required है. Destroying all forms of microorganisms and spores is sterilization, not asepsis. It is sterilization. Inhibits multiplication of pathogens. Pathogens रहेंगे but multiply नहीं करते and it will not cause infection in you. Inhibits multiplication pathogen in tissues is antisepsis. Okay. So asepsis is this absence of pathogenic microbes. McConkey agar. Tell me this one is a medium. Which medium? McConkey agar is which type of medium? Enriched है, enrichment है, differential है या synthetic है? तो McConkey agar is differential medium. It differentiates lactose fermenters from non-lactose fermenters. It acts as selective media, solid medium and indicator medium. McConkey agar can act as three mediums: selective, solid, because it is solid, and indicator. It indicates also. It is differential also. It is selective also. So three things are there. So McConkey agar is which medium? Differential medium. Enriched medium is example of chocolate agar. Is an example of enriched medium. Enriched medium is example of chocolate agar. What is the example of enriched medium? Chocolate agar. Enriched medium का chocolate agar. Differential medium, indicator medium, yes, selective medium, McConkey agar. McConkey agar. I hope you understood this. McConkey agar. McConkey agar. Okay, McConkey agar. So chocolate media is enriched medium. It is enriched with chocolate. Enrichment medias are liquid mediums, but this is a McConkey agar is a not enrichment medium. It is a selective medium, or you can say differential medium, or you can say indicator medium, because it is indicating whether it is lactose fermented or non-lactose fermented. Or you can tell it is a, or you can say it is a selective medium. Okay. Next question. McConkey is having three properties. One property is it is selective, it is solid, and other is indicator. What does it indicate? Lactose fermenters with non-lactose fermenters. Now this much is clear. Okay, now let's see 78th question. What is the question? Gamma radiation is used for sterilizing. Gamma radiation is used for sterilizing. Just now we studied, no? It is used for sterilizing plastic syringes. Not for cystoscopes, no. For cystoscopes, you use 2% Sidex. 2% Sidex you use. For dressing apron, for aprons, autoclaving you can do autoclaving. Metal instruments, hot air oven or autoclave. Hot air, or you can go for autoclave. The process of destroying all microbes including spores is called sterilization. It's called what? Sterilization. And destroying all microbes. Disinfection means you are just inhibiting dividing microorganisms. Inhibition of division, right? Antisepsis with tissues में नहीं होना, right? Inhibit multiplication of pathogens in tissues. Disinfection inhibits multiplication of pathogens on inanimate objects, right? Okay, out of the following, the true statements regarding sterilization is true statements regarding sterilization. Dry heat is the best method of sterilizing of liquid paraffins. For liquids and oils, you can use hot dry heat. All glass syringes are best sterilized by boiling at 100 degrees Celsius. No. Glass के लिए we use hot air oven. Bacterial vaccines best sterilized. No. By filtration. Pasteurization of milk by flash method is done by heating at 63 degrees Celsius. No. So for glass syringes you use what? Hot air oven, because they can bear hot temperature. Have you seen glass पूरा melt सा yellow orange color का? Bacterial vaccines, seeds filter. Use seeds filtration. Pasteurization of milk by flash method is at 72 degrees Celsius. For what? 15 to 20 seconds. By rapid cooling to 13 degrees Celsius. By then cooling to 13 degrees Celsius. 72 degrees Celsius for 15 to 20 seconds, followed by cooling to 13 degrees Celsius. That is called flash method. Flash सिर्फ 15-20 seconds में हो जाता है. It's like a flash. Now let's talk about these questions. Blood agar is an example of. So blood agar is an example of. I said na, blood agar, chocolate agar. These are example of enriched media. Then what is example of enrichment media? Selenite broth. Selenite F broth. Selective media, just now, TCBS agar or plet media, or you can say Akonki agar. Plet media. Okay, TCBS. Conditions required for autoclave is 121 degrees Celsius for 15 minutes. 121 degrees Celsius for 15 minutes. 121 degrees Celsius for 15. Sterilization of culture media containing serum is by. How can you do it? How can you do this? Culture media. Just now we discussed, na? Culture media sterilization containing serum is there. Then now you can do by inspisation. What is that method? Inspisation. So heating at 80 to 85 degrees Celsius for 30 minutes for 3 consecutive days. Used for protein-rich media like LJ and Dorset's egg media and serum-based Loeffler serum slope. Sterilization of fiber optic bronchoscope is done by glutaraldehyde. 2% glutaraldehyde. Least toxic irritant and is corrosive. Ideal for endoscopy and cystoscopes. 2% concentration for 20 minutes you have to keep. 20 मिनट के बाद ही वो स्टेरलाइज होगा. Glassware is sterilized by hot air oven. The disposable plastic syringes are commonly sterilized by ETO. Plastics by ETO, plastic syringes. Enrichment media. Enriched media is blood and chocolate, agar. Enrichment is selenite F broth. Chocolate is enriched. Selenite F broth, enrichment medium. The enrichment medium are usually liquid medium broth that allows the growth of certain microorganisms and inhibit other microorganisms. Selenite F broth, tetrathionate broth, and alkaline peptone water. So these are enrichment broth. They are liquid. Enrichment broth are liquid media. Selenite F and tetrathionate broth and the alkaline peptone water. Nutrient agar concentration of agar is nutrient agar. How much is the concentration of agar? 2%. Agar do not give any nutrition first of all. In nutrient agar concentration of agar is how much? 2%. Semi-solid agar, the agar concentration is 0.5%. Solid medium like nutrient agar concentration is 1.5 to 2%. Semi-solid 0.5. Okay. Blood agar is an example of enriched media. Is an example of enriched media and indicator media also. Indicator किस लिए? Step को करते ना. Okay. So Robert Koch assistant advised him to use agar instead of gelatin as culture media for cultivation of bacteria Robert Koch's assistant advised him to use agar instead of gelatin and culture media for cultivation of bacteria as gelatin melts at 27 degrees Celsius. So Robert Koch's assistant advised him to use agar instead of gelatin as culture media for cultivation of bacteria because gelatin melts at 27. So you cannot use gelatin as culture media. So agar is used as solidifying agent in culture media. It has no nutritive value. Advantage over gelatin are it is a bacteriological inert and it melts at 98 degrees Celsius, usually solidifies at 42 degrees Celsius, not unlike gelatin. Because gelatin melts at 27. So Robert Koch's assistant wife advised to use agar. Endoscope tube is sterilized by glutaraldehyde. I think 5th, 6th time this question came. By 2% glutaraldehyde you have to keep for 20 minutes. Next question, yes. Next question is this. Brownie's tube is used for steam sterilization. Okay. Brownie's tube used as a sterilization control for hot air oven and autoclave. For radiation, Bacillus pumilus, and filtration, Serratia marcescens. Okay, Brownie's tube used as sterilization control for hot air and autoclave. Prions are best killed by, how to kill prions? Autoclave plus sodium hydroxide, NaOH. Base. Prions are destroyed by autoclaving at 134 degrees Celsius for 1 to 1.5 hours. And treatment with 1 normal NaOH. Normality is 1 NaOH for 1 hour. Then only prions will be killed. Virus mediated transfer of host DNA from one cell to another is known as transduction. Transduction is the transfer of cell DNA by means of a bacterial virus. During the growth of virus within the cell, a piece of bacterial DNA is incorporated into the virus particle and is carried into recipient cell at the time of infection. This is called transduction. So virus mediated transfer of host DNA from one cell to another cell is known as transduction. So it is transfer of cell DNA by means of a bacterial virus, that is bacteriophage. We are using bacteriophage to transfer DNA. So during the growth of the virus within the cell, a piece of bacterial DNA is incorporated into the virus. Bacterial DNA is during the growth of the virus within the cell, a piece of bacterial DNA is incorporated into the virus particle carried into recipient cell at the time of infection. Bacteria may acquire characteristic by all the following except taking up soluble DNA fragments across the cell wall from other species. Yes, except बताना है. Incorporating part of host DNA, no. Through bacteriophages, yes. Through conjugation, yes. Conjugation, there is a sharing of there is DNA transfer, right? Yes, but not by incorporating host DNA. Okay, next is question is true about lambda phage is cause mad cow disease. No. Lytic and lysogenic interconversion contact. Lytic form incorporated within host DNA multiply. No. Lysogenic form incorporates with host DNA and remains dormant. So during the temperate or lysogenic life cycle, the lambda phage DNA remains integrated with the bacterial chromosome as prophage. Form incorporates with host DNA and remains dormant. It is not disturbing the cell. It is remaining dormant. Intra-species competition is the competition among individuals of a population. Individuals of a population. Intra-specific competition is the competition among individuals of a population. Individuals of a population. Intra-species competition. Individuals of the same population. Okay. Next question. Intra-species competition is an interaction in population ecology whereby members of the same species compete for limited resources. This leads to reduction in fitness for both individuals. By contrast, interspecific competition occurs when members of different species compete for a shared resource. Agar goat they are fighting for the grass, then it is intraspecific. Okay, but if goat and tortoise are fighting for the grass, then it will become interspecific. The following phenomena is responsible for antibiotic resistance in bacteria due to slime production. Not goat, but some goat and cow. Okay. The following phenomena is responsible for antibiotic resistance of bacteria. Slime production is biofilm formation. Repeat of the question. Shine Dalgarno sequence in bacterial mRNA is near AUG codon. The Dalgarno SD sequence is a ribosomal binding site in bacterial archaeal messenger RNA, generally located around 8 bases upstream to the start codon AUG. The RNA sequencer helps recruit the ribosome of the messenger RNA to initiate protein synthesis by aligning the ribosome with the start codon. Next question. Incorrect about plasmid. What is incorrect about plasmid? Involved in transformation. It is wrong. Incorrect about plasmid. So plasmid are involved in conjugation. They are not involved in transformation. They are responsible for multiple drug resistant capsule and pili formation. Okay, not for transformation but for conjugation. True about bacteriophage is they can transmit toxin to bacteria. No. They cannot transmit toxin like bacteria which transmit DNA to another bacterium. It is not a bacteria, it is a virus. Causes transformation of bacteria. Transduction, not formation. Virus which invades bacteria. Yes, this is correct. This is a virus. It can transmit toxin, gene, not toxin. It can not directly transfer toxin, but toxin gene that can give rise to toxin. It is a virus. It is not a bacteria. It is involved in transformation. Which of the following contains both DNA and RNA? DNA and RNA. Both DNA and RNA. Bacteria. Bacteria can contain both the DNA and RNA. Bacteria possess both DNA and RNA. Viruses either DNA, either Either DNA and RNA, bacteria both DNA and RNA. Prions they do not have at all. No nucleus, no nucleic acid. Horizontal transmission of R factor is by conjugation. Conjugation because it is through plasmid mediated. So R plasmids are transferred by conjugation, a horizontal gene transfer method. Carey and B. Mullis. Polymerase chain reaction was discovered by Carey B. Mullis. So PCR was discovered by Carey B. Mullis. Carey B. Mullis. Carey B. Mullis discovered PCR. F plasmid of high frequency recombination is F plasmid. F plasmid is for fertility factor. F for fertility factor. It is an extra chromosomal element. Integration with bacterial DNA episome. Integration with bacterial DNA episome. It is extra chromosomal combination. F plasmid of high combination is extra chromosomal, not in. It's extra, not intra. Okay, the extra chromosomal. Now, next question is false regarding the bacterial plasmids is. What is false? It is extra chromosomal. True. Eliminated by in treating with radiation. No, because it is part of the bacteria. Transmission of different characters. False regarding can cause lysogenic conversion. No, it is not true. Lysogenic conversion, it is true, means lysogenic conversion causes bacteriophage in the process of transduction. Plasmid can transmit multiple characters and responsible for toxin production, drug resistance, and virulence. It is false. They cannot cause. False बताना था ना. Okay, false regarding bacterial plasmids. So yes, if bacteria will die, plasmids will be gone. Extra chromosomal transmission of different characters like drug resistance, virulence, toxin production. Lysogenic conversion caused bacteriophage is the process of transduction, not by plasmids, by bacteriophage. Drug resistance transfer by bacteriophage. Drug resistant transfer by bacteriophage involves. Bacteriophage is transduction. Among gene transfer process, bacteriophage is involved in transduction. It is transduction. It is inducting something. Okay, inducing. It is going inside the bacteria. Okay. True about transferable resistance. High degree of resistance, yes. Involves resistance to multiple drugs. Plasmids play a role. All the above are true. No use of dose adjustment combinations if they are resistant. And if they are resistant, they will not respond to dose adjustments. If you increase the dose, then also they will show resistance. Horizontal transfer is there, and there is no change in virulence. Phage typing is useful as an epidemiological tool in all except phage typing, except Shigella. Shigella is a epidemiological tool for Salmonella, Vibrio, Staphylococcus, not for Shigella. Next question. Drug resistance is not transmitted by. So drug resistance is not transmitted by. So drug resistance, it is not transmitted by chromosome. It can be by HFR, plasmids, transposons, but not by chromosomes. Following transmit drug resistance except what? HFR. HFR is fertility. Okay. All other choices are transmit drug resistance. All are true regarding plasmids except following transmit drug resistance except HFR, right? All are true regarding plasmid except involving multi-drug resistant transformation. No, not for transformation. Conjugation. Plasmid बोले तो conjugation. PC Remember Priyanka Chopra, PC, plasmid conjugation. Imparts capsule formation, imparts pili formation, yes. Multiple drug resistance, virulence, all that. Okay. Conjugation does not involve bacteriophages because conjugation bacteriophages is transduction. Involves pili, capsule, plasmid. Bacteriophages are involved in transduction. Jumping gene is nothing but transposons. A transposable element or transposon is a DNA sequence that can change its position within a genome, sometimes creating or reversing mutations and altering the cell genetic identity and genome size. Microbial killing below MIC, minimum immunity minimum concentration, is known as post antibiotic effect. The post antibiotic effect, PAE, is defined as persistent suppression of bacterial growth after a brief exposure of 1 or 2 hours of bacteria to an antibiotic, even in the absence of host defense mechanism. Factors that affect the duration of the post antibiotic effect include duration of antibiotic exposure, bacterial species, culture medium, and class of antibiotic used. It has been suggested that an alteration of DNA function is possibly responsible for post antibiotic effect, following the observation that most inhibitors of protein and nucleic acid synthesis, aminoglycosides, fluoroquinolones, tetracyclines, clindamycin, certain newer macrolides, ketolides, and rifampicin and rifabutin induce long-term PAE against susceptible bacteria. Now let's talk about transformation. Mechanism of direct transfer of free DNA. Direct transfer is what? Transformation. Transduction, bacteriophage. Plasmids, conjugation. Transformation is the process by which a bacterial DNA molecule is taken up from the external environment and incorporated into the genome of the recipient bacteria. External environment it is directly taking. Mechanism of direct is through the transformation. This taking from the environment and getting transformed. Endotoxin transfer of some extra chromosomal material from one bacterium to other can occur via F factor, C factor, R factor, all the above. F factor is fertility factor, C for colicin production, and R for resistance. Okay, endotoxin from gram-negative organism is what I already discussed. That is lipopolysaccharide. Lipopolysaccharide, also known as lipoglycans and endotoxins, are large molecules consisting of a lipid and a polysaccharide composed of O antigen outer core, inner core joined by a covalent bond. They are found in the outer membrane of the gram-negative bacteria. Next question: the term viable not cultivable, VNC, is used for. Viable not cultivable is used for. The term viable not cultivable, VMC, is used for lepra. Lepra is not cultivable and it is not viable outside, right? Because the intracytoplasmic. And Treponema pallidum, syphilis. So other VNC like chlamydia, rickettsia, and viruses, they are also okay. Understood. So these are the chapter 1 general microbiology questions. I hope you liked it. So let's discuss the next chapter in our next video. Thank you so much. If you have been through the video, if you find that these videos are making your life easy, then please hit that subscribe button and like and share with your friends. Thank you so much. Don't forget to like, share, and subscribe.